Okay, welcome back to the Chapter 9 lecture of Patterns of Inheritance. This will be part 2 of Chapter 9. So I was mentioning Mendels Law of segregation. What that means is we all have two alleles, two forms of a gene for each individual locus or location on a chromosome. So when our sperm and egg cells are developing, what that means is the allele pairs that we have inherited from our parents are separated from one another. And that allows for our sperm and egg cells to get basically half the amount of genetic material that our normal body cells have. So what this is showing here is we have our representation of chromosomes here. Let's say the yellow one came from the mother and the blue one came from the father. And of course, in real life, for humans, we have 23 pairs rather than just a single pair of chromosomes. But just for the purposes of illustration, it's just showing a single pair of homologous chromosomes. So in interphase, prior to meiosis, we have the creation of sister chromatids, which are identical genetic copies of each chromosome. So that's what this is showing. And then this is showing the result after meiosis 1. We have the separation of homologous chromosomes. And then this is showing the result after meiosis 2, the separation of the sister chromatids. So as with this original pair here, we would have, say, an allele here, and then a corresponding allele here for a single trait. Now you can see that after meiosis, we have separation of those allele pairs. So we have the one allele here, and that's in a single sperm or egg cell. Once we have fertilization occurring, then you again have those matching allele pairs. Could, of course, be a different gene variant depending on the genetics of the partner. So let's talk about dominant versus recessive and what that means. Dominant alleles determine the organism's appearance in the case of these traits that are just controlled by a single pair of alleles. So we'll see many different examples of different patterns of inheritance. But for now, we're just talking about the traits that Mendel studied extensively and observed, and these are simple dominant recessive inheritance pattern traits. So a dominant allele determines the organism's appearance. They are expressed in the phenotype, and they can also be said to mask the effect of the recessive alleles. And in a genotype, they're always going to be written in the form of a capital letter. So this right here would be a dominant allele. This would be a dominant allele. That would be the allele that gives the pea plants their purple flower color. This, however, the little p here would be a recessive allele. So the recessive alleles have no noticeable effect on an organism's appearance if they are paired with a dominant allele. They are, in fact, expressed in the phenotype when no dominant allele is present. So if we only have the presence of recessive alleles, say this gamete has the recessive allele here, and it pairs up with a gamete that also has that recessive allele, you'll notice we have this white flowered pea plant here. That would be the result of two recessive alleles coming together. But if a recessive allele is paired with a dominant allele, that dominant allele is always going to mask the effect of the recessive. So you'll get a purple flowered plant instead of a white flowered plant. And of course, these are written in the genotype as a lowercase letter. Now here's a couple other terms for you, and this has to do with whether an organism has identical alleles or different alleles for a single gene. Homozygous is when an organism has identical alleles. So they've received the same gene variants for a single gene at a single locus on a chromosome. So for example, big P or little p, doesn't matter if they're dominant or recessive, if they're the same, if that organism has received the same gene variant, it's considered homozygous. If an organism is heterozygous for a particular allele, then that organism has different alleles for a gene. So for example, again, if we're looking at homologous chromosomes here, homozygous, big B, homozygous, little b, heterozygous. In either order, you just have to have one of each, big B, or little b, big B. So now we can see how Punnett squares can come in handy. We can predict outcomes of crosses between organisms for traits with simple dominant recessive inheritance patterns. A Punnett square is just a diagram that illustrates all possible outcomes from various breeding scenarios. And again, this is only going to be for monohybrid crosses. Crosses between organisms that differ visibly in just a single trait. So in this case, we have T, big T for tall, little t for short. This is the genetic basis of how tall a plant can grow. And in this case, big T is dominant, little t is a dwarf form of the plant, and it's short. So what this is showing, these would be the possible contributions from a single parent for a single gene variant at a single locus. And then these would be the possible contributions from the other parent at the corresponding locus on the chromosome. So everybody, of course, has two alleles for each gene locus. You receive one from each parent. When our gametes are created, those are going to separate from one another. So you can say that 50% of the gametes will have this allele, and 50% of the gametes will have that allele, and then the same holds true for the other parent. So what this is showing, and this first one isn't actually a great example because they're all the same, but what this is showing is the possible offspring for this one particular trait that this pair of parents can produce. So remember, keep those axes in mind. These would be the alleles for, say, the father. These would be the alleles for the mother. How they combine within this Punnett square tells you what the offspring can have. So in this case, we only have big T, we only have tall plants. The only thing that can result from that is homozygous tall plants. Similarly, if we look at this example with little t, short plants, the only thing that can result is short offspring. Now, what I would like you to do is take a minute to think about how this works and try to see if you can come up with the results for this Punnett square here. Okay, I'm going to go ahead and go over this, so feel free to pause if you want a moment to think about this. But again, how we do this is we drop down this big T and we carry across that big T. So in this case, we have one offspring box taken care of. That represents 25% of the offspring. So the whole box represents 100%, and each little box represents 25% of the offspring. Okay, so again, drop down the little t, carry across the big T. Just by convention, the dominant allele is often written first, but you can do it either way, whichever helps you keep track of it better. Same, drop down the big T from up here, carry across the little t there. Over here and here we have just little t, little t. There. So that will tell you that 25% of the offspring from this particular cross is going to be homozygous, dominant for that trait. 50% of the offspring are going to be heterozygous, also going to be dominant because we have one dominant allele, and that's going to mask the effect of the recessive allele. And then 25% are going to be homozygous recessive. But since there's no dominant allele, they will actually have that short height. Okay, let's do this other one here. Big T, big T, big T, big T, big T, little t, big T, little t. So in this case, are there going to be any short plants? There's not going to be any short plants because there's at least one dominant allele in every single one of these boxes. So that's basically how a Punnett square works. Okay, let's do some more practice with these. Draw the Punnett square that would result from a cross between parents with genotypes big B, little b, and big B, big B. And then answer these questions. So go ahead and pause if you would like. Take a minute to think about this, and I will go over the answers shortly. Okay, big B, little b, and big B, big B. Whoops. Just when I thought my tablet was cooperating. This is more going to be like a Punnett parallelogram rather than a Punnett square, but okay. So we have big B, big B, big B, little b, big B, and big B, little b. Okay, so what percentage of the offspring will be homozygous dominant? So homozygous refers to genotype, dominant refers to phenotype. Homozygous dominant would be this box and this box. Add those together, 25% plus 25% is going to be 50%. What percentage of the offspring will be heterozygous dominant? Again, heterozygous refers to genotype, dominant refers to phenotype. So it's going to be 25% here plus 25% here equals 50%. What percentage of the offspring will be heterozygous recessive? Now, I usually am not one to write any sorts of trick questions, but this is kind of a trick question. So in the case of simple dominant recessive genetics, is there such a thing as heterozygous recessive? No, because you have to have two recessive alleles in order to have the recessive trait. So heterozygous recessive, that doesn't even make any sense here. It would be 0%. Okay, let's talk briefly about dihybrid crosses. Sometimes I like to put a dihybrid cross as extra credit on my midterm exam. And I think I've had one person get it correct in the several years that I've been teaching this course. It's always fun to see what folks come up with. Now, the tricky part about this is you have to account for both gene loci. And a lot of people forget that. A dihybrid cross is the mating of two organisms that are alike except in two characteristics or traits. So it's like running two monohybrid crosses simultaneously. So instead of just a single pair of alleles, you have two pairs of alleles. So what this is showing here, we have this white bobtailed cat and a tan long-tailed cat. What this is showing is in the F1, you can tell that tan fur color is dominant over white fur color. You can also tell that a short tail is dominant over a long tail. And then only in the F2 do you get this interesting recombination effect where you see cats with the parental phenotypes as well as combinations of those phenotypes. So tail length and fur color are recombining in various ways.